Thank you very much. That was a good segue into my presentation. I have no conflicts of interest to disclose. So, cancer arises through as a result of through a multi-step mutagenic process, and through genetic mutations. And by mutations, I'm not just referring to the inherited predispositions that we just heard about, but specifically acquired genetic mutations that give rise to de novo malignancies. These mutations occur in a common set of hallmarks in order to bring about a malignant phenotype through one variation or another. And the mutations occur in the form of different point mutations or chromosome translocations, deletions, amplifications, and epigenetic changes. In the majority of cases, obviously, the cause of these mutations is unclear. And in many cancers, certainly there are genetic mutations that have been identified, but it's not always quite clear whether or not these mutations are simple associations versus direct causation. CLL falls into this category as well. CLL is the most common form of leukemia in the Western world. It's a malignancy of mature CD5 positive B cells with an incidence that rises linearly after the age of 50. How's this pointer work? There we go. It has a median survival of about 10 years, and as such, it's sort of generally believed that the majority of patients will have a rather indolent course and generally die of causes unrelated to their CLL. However, this is true for less than 30% of patients, and a significant number of patients die within 1 to 2 years from complications of CLL, while others will live 5 to 10 years with a relatively benign course, followed by a terminal phase lasting 1 to 2 years. From a genetic standpoint, as far as figuring out what the causes of CLL are, there are a number of cytogenetic abnormalities that are associated with the disease. And many of these have significant prognostic importance. 13q abnormalities are the most common deletions in CLL and confer a favorable prognosis with a median survival of about 10 years or more. In contrast, deletions of the short arm of chromosome 17 are the least favorable, and with a survival of less than 3 years. Now, about 82% of patients have clonal cytogenetic abnormalities associated with their disease. But in many cases, the relevance of the mutations is unclear. Chromosome 17p deletions, as I said, are associated with shorter survival and also appear to have a diminished response to therapy, specifically fludarabine, which has been one of the staples of CLL chemotherapy for many years. One of the genes on 17p encodes p53, and about 30% of patients with CLL will have p53 mutations independent of 17p deletions. And it's thought that p53 mutations may be related to chemo resistance in this subset. Patients with 11q deletions often have lymphadenopathy and rapid progression of disease. One of the genes on 11q, at 11q23, is ATM, which leads to impaired DNA damage response, although it's unclear exactly what the contribution of that is to the pathogenesis of the disease. Trisomy 12 is relatively common in CLL with intermediate survival, but patients often present with advanced stage disease. The molecular association with trisomy 12 remains undefined. 13q deletions, as I mentioned, are the most common and most favorable, and have attracted a lot of attention as of late because of the identification of several microRNAs within the minimally deleted region. And these microRNAs apparently target repression of BCL2, which is an anti-apoptotic survival factor. And so the deletion of these microRNAs in this subset of patients may lead to overexpression of BCL2 with enhanced survival of the cells. Most recently, there was a a study in Cancer Cell describing a specific targeted deletion of these microRNAs and the minimally deleted region in the B cell lineage, and these mice developed both CLL as well as a non-Hodgkin's lymphoma, again supporting a possible role for these microRNAs in the pathogenesis of the disease. It should be noted, though, that these deletions encompass megabases of DNA, and so there are probably a whole host of other genes involved that have yet to be discovered or characterized. Now, at the molecular level, there are a number of abnormalities associated with CLL, including about 25 different microRNAs that have been described as dysregulated. Gene microarray technology generates data faster than it can be analyzed, and so although there have been a number of genes that target a whole host of factors, including cell cycle and proliferation, apoptosis, and DNA damage response, a specific role for many of these microRNAs is yet to be established in the pathogenesis of CLL. TCL1 is interesting because it suppresses apoptosis and increases NF-kappa B dependent transcription, but it's not normally expressed in mature B cells. It's normally expressed early in B and T cell development, but high expression has been observed in patients with unfavorable risk factors, and in some series it's been shown to be an unfavorable risk factor in and of itself. Perhaps the most studied factor in CLL has been the B cell receptor. B cells produce immunoglobulins, or antibodies, and express surface immunoglobulin in the form of the B cell receptor. And mutational status of the immunoglobulin heavy chain gene is a strong independent prognostic indicator. So what do I mean by mutational status? So the humoral immune system is capable of recognizing and clearing virtually any invading pathogen that our body encounters. And this is made possible because our B cells produce a huge diversity of antibodies that are capable of recognizing their target antigen. And so the germline immunoglobulin gene in primitive B cells is composed of hundreds of variable diversity and joining regions. And during early B cell development, these V, D, and J regions undergo a process known as VDJ recombination. This is an antigen-independent process that goes on in the bone marrow. And what happens is individual variable diversity and joining regions are assembled to form a mature immunoglobulin gene. This immunoglobulin gene is then expressed in naive or immature B cells as a surface antibody. Then, upon encountering an invading pathogen, B cells undergo a second round of diversification whereby point mutations are introduced into the variable region. And these point mutations result in an antibody that has increased affinity for its target antigen. So basically what you have are two subsets of patients in CLL as far as mutational status, those that have unmutated variable regions and those that have mutated variable regions. And so, as I suggested before, Ig heavy chain variable region, or IgVH mutational status, is a strong predictor of survival in CLL, with unmutated CLL patients having a median survival of about 8 years or less, and mutated IgVH CLL having a median survival of about 15 years. And so this raises the question of, does this tell us anything about the CLL clone, or the origin of the CLL clone? Because so by default, naive or pre-germinal center B cells have an unmutated variable region. They encounter an invading pathogen where they enter the germinal center response of lymph nodes, and through a T-dependent process, these cells undergo rapid proliferation and expansion. During this step of differentiation, these central blast B cells undergo somatic hypermutation of their variable regions. They then further differentiate into centrocytes, where they undergo class switching in the light zone, and selection of cells that have high affinity for their target antigen. At this stage, you have memory B cells and plasma cells with mutated variable regions. And so the thought is that patients that have unmutated IgVH represent a pre-germinal center phenotype, and those with mutated represent a post-germinal center phenotype. However, it may not be that simple. Now, to look at mutation status is quite labor-intensive and cumbersome. It involves cloning and sequencing, and is not done routinely except in the setting of clinical trials. And so there have been a number of studies to try to identify surrogate markers of mutational status. Most notably, ZAP70 and CD38 both have been identified as being highly expressed in patients with unmutated IgVH, or poor prognosis patients. Interestingly, if you take B cells from patients in culture and stimulate them in vitro through the B cell receptor, patients with unmutated IgVH seem to have enhanced B cell receptor signaling, while patients with a mutated IgVH have diminished B cell receptor signaling, and that when you stimulate these cells, they actually just become anergic. And the thought may be that the presence of ZAP70 and CD38 enhances signaling through the B cell receptor. There also seems to be some bias in V gene usage between unmutated and mutated CLL, and that there's often a preference for V169 or 321 in unmutated, or V434 in mutated CLL. As I mentioned, TCL1 is overexpressed in unmutated CLL as well. Now, an interesting observation is that all CLL, mutated or unmutated, seems to have a immunophenotype of an antigen-activated B cell. And by gene expression profiling, both subsets resemble memory B cells. And so the idea that these unmutated B cells may represent a pre-germinal center B cell may not be entirely correct. One interesting aspect of this is that patients with unmutated variable regions also express the protein AID, or activation-induced cytidine deaminase. And so AID is the protein that is responsible for somatic hypermutation and class switching. It introduces point mutations into the variable region by converting the cytosines to uracils. These are then repaired in an error-prone manner to give rise to point mutations, and that's what contributes to affinity maturation of antibodies. AID also introduces double-stranded DNA breaks into the switch repeats that flank the constant regions. These double-stranded DNA breaks are then processed, and then non-homologous end joining results in class switching. So AID is expressed in germinal center B cells almost exclusively. It's essential for somatic hypermutation and class switching in that mice that are deficient in AID don't switch and they don't mutate. But unlike VDJ recombination, which is a site-specific process that requires the presence of recombination signal sequences that flank the VDJ regions, there doesn't seem to be any real sequence specificity to AID targeting. And the mechanism that guides AID to its genetic target is entirely unknown. So as such, AID is a DNA mutator that represents a threat to genomic integrity. And in fact, there have been a number of studies that suggest that AID activity may actually contribute to point mutations and translocations of a number of genes that are associated with B-cell malignancy. Now, David Schatz gave a talk in Cancer Center Grand Rounds a few weeks ago, and he presented some of this data. But just to show you quickly, I've been working in his lab, and so I have access to this. Maggie Liu is a graduate student in the lab, and she found that simply by looking at B cells from normal wild-type mice, obtained from germinal centers, that approximately 25% of the expressed genome accumulates mutations as a result of AID activity. This is in contrast to AID knockout mice, which don't seem to accumulate mutations to any significant degree, and this background mutation frequency of 1.6 is probably due to the sequencing procedure itself. And so this raises the possibility that the question of, if you have a B cell that expresses a DNA mutator, is AID possibly playing a role in the pathogenesis of B cell malignancies, specifically CLL? And so there have been a number of small studies looking at expression of AID. In this example, what has been shown is that indeed AID is expressed in patients with unmutated IgVH regions, but invariably it's not expressed in patients with mutated IgVH. Now, at a quantitative level, the level of AID expression seems to be quite low compared to that of germinal center B cells, and it also seems to be quite variable. However, given that we also know that there's enhanced B cell receptor signaling in these patients that have unmutated IgVH status and low expression of AID, is it possible that during the course of the disease that AID expression may vary or may be stimulated in the microenvironment? And so I did a very straightforward experiment and just looked at CLL cells from a cell line in several patients with unmutated and mutated IgVH status, and found that first I confirmed that in untreated cells, yes, there is low expression of AID, but if you take these cells and you stimulate them with either through anti-IgM and IL-4 or through CD40, you get very high expression of AID. Yet in patients with unmutated IgVH, the levels of AID go up only minimally. And so this raises the next question of, really what is the direct role of AID, if any, in the pathogenesis of CLL? And so in a collaboration with John Bird at Ohio State, we've used the emu TCL1 transgenic mouse as a model of human CLL. And so these mice, as I said before, TCL1 is overexpressed in a subset of patients with CLL, specifically those patients with unmutated IgVH. And in the TCL1 transgenic model, these mice actually develop an oligoclonal circulating CD5 positive, IgM positive B cell hyperplasia that starts at about 3 to 6 months of age. By about 8 to 16 months, a monoclonal B cell population emerges with involvement of bone marrow, lymph nodes, and spleen. Now, interestingly, like the aggressive form of human CLL, the CLL cells from emu TCL1 transgenic mice develop unmutated IgVH, high expression of ZAP70, and I've shown that they also express AID. So our hypothesis is that AID is involved in the pathogenesis of CLL, and our simple experiment was to just cross the TCL1 mice onto an AID deficient background. And what we see is that emu TCL1 mice have normal B and T cell development and normal development of lymphoid organs. They develop, after a latency period, an oligoclonal CD5 positive, IgM positive, CD11B positive population. And AID knockout mice also have normal B and T cell development and normal development of lymphoid organs. They do have enlarged germinal centers, but they have complete absence of somatic hypermutation and class switching. And so very preliminary data from this, as this has been ongoing, and we've just gotten our first cohort of mice, is that, as expected, the emu TCL1 mice develop an expansion of CD5 positive, IgM positive cells starting at about three months, and AID knockout mice don't seem to develop this population. However, the mice, the TCL1 transgenic mice that are on the AID knockout background seem to have a delay in the expansion of this population. Now, as I said, this is very early data, and the investigations are ongoing to further characterize the B-cell compartment in these mice. But for now, our thought process is that perhaps AID is somehow contributing to the pathogenesis of CLL, at least in this model, perhaps either through introducing mutations into genes that are important for proliferation, or through introduction of mutations into genes important for cell survival, or both. And so just to summarize, CLL may represent multiple clinical subgroups based on cytogenetics, but two biological subgroups based on IgVH mutational status, as well as response of B-cell receptor to signaling. Unmutated IgVH CLL may originate from an early germinal center B cell rather than a pre-germinal center B cell, with competent B cell receptor signaling and expression of AID. AID is a DNA mutator that is responsible for somatic hypermutation and class switching of the Ig genes, but is also capable of introducing mutations throughout the genome. AID may play a role in the development of CLL in the TCL1 transgenic mouse model, but further investigation is needed to determine the clinical and biological significance of AID in the pathogenesis of CLL. So, big picture, future directions of this work is in the future, hopefully, as technology becomes less expensive, whole exome or whole genome sequencing may actually provide more information about the specific genetic mutations and what mutations we're missing that might be involved in the pathogenesis of this disease. In addition, as technology permits more advanced epigenetic profiling, I think we'll probably find that there's a significant number of epigenetic changes that may also be involved. Familial CLL is somewhat rare, but may provide a path to identify some clues to genes that may be involved in sporadic CLL as well. In addition, future studies on B-cell receptor signaling and competency in these cells may be important for identifying targetable factors, specifically ZAP70, potentially. In addition, looking at the role of the microenvironment in the pathogenesis of the disease will become important. And then finally, sort of the gratuitous statement is that some of this work hopefully would eventually lead to the identification of potential targets for therapeutic intervention. So I'd just like to acknowledge David Schatz, whose lab I've been working in, and some of the other members of the Schatz lab that have been helping me on some of this work. My collaborators at Ohio State, John Bird and Amy Johnson, and my funding sources. Thanks.